Hello, my dear ones. Yes, tomorrow is a biology practical, and I believe you already know all the specimens you'll be treating, you'll be answering questions on tomorrow. So, I want to talk on one of the specimens, the fresh milk. What if you are asked to carry out burette test on the fresh milk? What do I mean by the burette test? You may be asked to add 1 or 2 ml of sodium hydroxide plus 1% of copper tetrahydrosulfate 6 or CuSO4 into your what? Fresh milk. So that's the test. What will be your observation? The observation will be what? Violet or purple color. Okay? It's observed. Then your inference will be what? Protein is present. Then you can be asked that what name can be given to this test. So see, the simple answer is what? A burette test. That's the test that we call burette test, whereby sodium hydroxide and copper tetrahydrosulfate 6, the two are added into what? A protein food, a proteinous food, to give us a violet or purple color. That is for the milk then. Now, what of the thermometer, the quadrat, the meter? All these instruments, they are ecological instruments. So you can be asked that what branch of biology do we use this instrument for? So your answer is very simple. The answer is what? Ecology. Now you can also be asked that how do you define ecology? Ecology is just what? A branch of biology that deals with the study of living things in relation with their environment. So what do we use this ecological instrument for? Your quadrats, we use it for what? For sampling under ecological study. Then you have your meter for measuring. Okay, we use your meter for measuring, and the thermometer to measure what? The temperature or the hotness or coldness of what? Of an environment. Very simple. What if you are not asked to draw? Very simple. When you're asked to draw in biology, all you need to do is to measure. Just measure. Measurement, that's your size. Ensure that you use your ruler to rule the guideline. Very, very important. And ensure that the guideline touches the part that you want to what? You want to label. Then don't forget your magnification. Magnification is very, very important. Put your magnification there. Make sure you measure, that's the size. And make sure you give your drawing an appropriate what? Title. And you put all the necessary labelings on your diagram. With that, you are good to go. Okay? Then concerning your specimen A and specimen B, where you have groundnut pod and you have your roasted groundnut seed, okay? Please do not forget to put the seed there. Don't just write groundnut. Groundnut what? Groundnut seed. That is your specimen B, roasted groundnut seed. Not putting the seed there can what? Can make you to lose the what? The mark. And specimen A, your groundnut pod. So you can be asked to state the observable differences. What do I mean observable differences? That means what you can see. One is a pod, one is what? Simply what? Just seeds, okay? Then one, the shape of the pod, you can see that it's elongated compared to the shape of the what? Of the specimen B, which is, and in terms of the color as well, the roasted groundnut seed, you can see that one is brownish. And the pod, the groundnut pod, you can see that one is not what? Brownish. So in terms of the color, that's another observable differences. Then in terms of the size, that's another observable what? Difference. Those are the things you need to what? You need to note. Or you can be asked that what fruit type today they are legumes, okay? Groundnut seed or groundnut pod, both specimen A, specimen B, they belong to what? To the legume. Very simple. Then you can be asked that what is the nutritional value? The nutritional values are what? They provide us with what? Protein. They provide us with protein. That is for your specimen A and your specimen B. Okay, so what if you are not asked to crush your specimen B on a filter paper, state the observation and the inference? Very simple. If you're asked to crush, just crush on your filter paper. You observe what? A translucent what? Appearance. A translucent paper, a translucent filter paper. Then your inference will be what? Oil is present, or fat and oil are present. Okay, oil is present, or fat and oil are present. Very simple. Okay, so for your what other specimens do we have there? You have the what? The alimentary tract of a small mammal. You can be asked what is digestion. You can be asked to state the alimentary tract. Name all the parts of the alimentary tract of a small mammal. Let's say for instance rats. Okay, you will observe, you see the stomach there. Okay, you see the intestine, small and large intestine there. Okay, so all these things you need to read. I want my student to read. Okay, read on this part, read on digestion. Okay, the role of each of these parts of the alimentary canal in rat to be specific. Okay, then when we are talking of your what again, tell me. Okay, if anyone, maybe you follow me for more tips. Okay, all right, please, I want you to work on all the things I've mentioned. I wish you all the best. A1 is surely yours. Bye.